listening to the previous speakers, I've kind of, you know, it put some ideas in my head about where we currently sit with regard to this. You know, Jackie called me out earlier because I was giggling at one of her slides, and the reason I was giggling was not because of what she was saying. It was, I think I had a realization, if we think about everything that's going on in terms of genetic and environmental influences and everything else, we're closer on her little Venn diagram to anarchy than we are to the simple solution. So that's why I was giggling back there when I saw that. Right. So anyhow, so what I was asked to do today is talk about the genetic prediction for bovine congestive heart failure. This is work that we've undertake as a result of funding we received from the Foundation for Food and Animal Research and their ICASA program. So I just wanted to mention that right up front before we get going. So if we think about, what I'm going to talk about today, and the idea of a genetic prediction. So a genetic prediction for heart failure, that's really something that's relatively new, right? And so, I thought it might be good to just sort of outline what I think about when it comes to what do we need for a useful genetic prediction? Like how do we arrive at the end point of something that you can select or make selection decisions on? So, the first thing is really, can usable data be found, right? And so with genetic evaluations, one or two observations, we really, they're not that useful, right? So we need thousands. We need a lot of data that we can collect relatively cheaply, relatively inexpensively, get that reported, get the pedigree associated with that, and then we can actually come through and make a genetic evaluation. So the usable data is a real big piece to the development of an EPD. So variations, the next one on here, right? So if all the observations were the same, then we can't have we can't differentiate between one animal or another, whether one animal's better than another. So we really can't make any sort of decisions based on an animal's merit if all of the observations are the same. So it's got to be heritable, right? And then lastly, I have on there economically relevant, okay? So, and I'm not talking about, the classic definition of an economically relevant trait. This is more of, does the trait influence profitability, but not only for itself, but what is its relationship with all the other suite of traits that are currently published on breed association websites, right? There's a whole host of traits. So to me, the big key here is the background work on understanding the consequences of selection on existing suites of EPDs, right? And so I'm going to talk about all that as we move forward. So in terms of data, and I know Dr. Holt had some pictures of these up earlier, and then Dr. Vanderlei had this up as well. But this is the scoring system that we are currently using to collect heart failure data, right? So this scoring system ranges from a 1 to 5, right? A 1 up there on the very top left part of your screen is the normal heart. The number 2 is another version of normal. So I'm not exactly sure what another version of normal means, other than the fact that it's used a little more and it's stressed a little more, but it's not so far that it's really having deleterious consequences on the individual. But Dr. Holt says 1s and 2s are normal, 3s, 4s, and 5s are a problem, okay? So this is the scoring system that we use. And in terms of collecting data, this is something that we can go into a beef plant, watch them go along the conveyor belt, one every 5-6 seconds, and we can just sit there and score hearts. So we can collect these at a very quick pace. It's relatively cheap. There's not a whole lot of technology involved. It's just paying for someone to go stand there by the stand there by the table and as these animals are going by and just scoring those hearts. One thing I do want to mention though is, so we've got this range of 1 to 4, and you'll see, or 1 to 5, but you'll see a difference on what we're observing here on the next slide with regard to those 5 hearts. So in terms of variation, the first thing that we did in this project is we really wanted to understand, what is the relationship between heart score and everything else that we're already measuring on these animals. So Dr. Vanderlei talked about, I forget the term he used. I'm trying to think of it. Essentially alive or dead, right? We have a failure in the feed yard versus animals that didn't. So there is a distribution of heart scores under that sort of those extreme endpoints or that extreme umbrella. So one of my biggest goals was to really understand how heart scores relate to the other carcass traits, how they relate to feed intake, how they relate to pulmonary arterial pressures that we're already measuring. So I'm gonna show you some results from quite a few different pieces or components or data pieces that we've collected. So a little bit about our data. So this, right now, this is data that was in the first year of this 3-year project. So currently we have about 1,400 head of cattle, and these were all Angus influence breeds that were born, raised, fed, and harvested out in the panhandle of Texas. So we collected heart score, carcass data. There's a subset of animals that have pulmonary arterial pressure measures. There's a subset of animals that have feed intake measures, so measures of efficiency. And then we took those, collected that data, spent a lot of days on the road driving through snowstorms, trying to get down to Amarillo to collect hearts at the plant. But in summary though, if we look at heart scores, right? So this is the distribution of the heart scores that we have found in this data set. So again, we have the 1s and 2s, right? That's the majority of our observations. And so these are just animals that have gone through, made it to harvest, scored their hearts. So just doing the quick math on there, that's 71%. So 71% of the animals have normal hearts. But there's 29% of those in this population that had heart scores that we would call unhealthy or 3s or 4s. One thing you'll notice here is that we have no 5s in our data set, right? And so the feeder that we were working with, they didn't really have any mechanisms of realizing animals prior to them having heart failure, or what I call extreme cases of heart failure. So we never observed those in the actual plant. Now this 29%, you might think, well, is that high or low? And I'm gonna say it's gonna depend on the population, I think, and depend on the makeup of the populations, whether, the crossbreeding and everything else that goes along with those. But, there are certain populations where we're seeing incidence rates up closer to 40% of the cattle coming through with some form of affected or unhealthy heart at harvest. So that then kind of asks the question, well, if they're experiencing heart failure, what does that do to everything else that's going on that we're already getting paid for? The carcass characteristics, that type of thing. So this is a slide of summary statistics, and I have this up here. I'm not gonna walk through all these because I'm not, we can get into the nuts and bolts and the details. But really what I wanted to point out is in addition to heart score, we've collected serial PAP measures. So we PAP them early, 9 months right as they're entering the feed yard. And then we're PAPped a group of them again, sort of, a few weeks, 2, 3, 4 weeks right before harvest. So that's that 9 and 14 month PAP. So you're gonna hear me refer to the PAP measures as we move forward with regard to 9-month or 14-month or late PAP. But we've got our carcass measures, back fat, marbling, all the way down through average daily gain, feed intake, and there's even a sparse amount of weaning weight data in there, although that's you know, I'm not too comfortable on how reliable that data is, at least from this study. All right, so let's look at relationships here real quick. And so this is just a phenotypic correlation heat map. And so what correlations do is they detect linear relationships between two variables, okay? So the higher the correlation, correlations range from -1 to 1. So a correlation near 1 or -1 is very strongly correlated. A correlation near 0 means there's no relationship, okay? So up here, the red colors indicate a positive relationship, meaning as one is going up, the other one's going up. And the blue is a negative relationship, meaning as one is going up, the other one's going down, okay? So if we look at this, just this heat map, you know, you see up there in the top right corner, there's some very dark red boxes, okay? And those are all the pulmonary arterial pressure measures, and those are all highly related. But if we look at that last column, so that very last, that high column right here, that's the heart score where the font is cut off a little bit. Up here on the very top corner, that's our heart score column. And every block or where it intersects on a row, that's going to be the level of the correlation. So what this does is it kind of gives us an idea on strongly related versus sparsely related in terms of their linear relationship between two traits. So for instance, if we look at, and because my eyes are bad, I'm going to look here at the slide real quick. If we look at PAP 14 versus heart score, there's a strong, sort of moderately strong positive relationship there. And then if we get down to things like marbling, there's really not much color there, so there's really not much of a relationship, right? So does that mean there is any relationship? No, because this is testing whether that relationship is linear. And so we're going to get into what that means here on the next few slides. Okay, so let's talk about PAP first, right? So we're kind of picking up on where Dr. Enns left off. So these are the distributions of the pulmonary arterial pressure measures that we recorded on these animals at 9 months and at 14 months, right? So on the left chart, we have the 9-month PAP, and as you'll see, it's a very tight distribution of PAP measures. And actually, there are really no animals in this population at 9 months of age that we thought were a problem, right? And so if you would take our Rouse data, for instance, at high elevation, you would see a much bigger distribution in terms of pulmonary arterial pressure. But these animals are at low elevation, right? They're not being stressed. They're young animals. They're not being pushed in the feed yard real hard just yet. So there is no heart problems as of yet, at least in this population, from their pulmonary hypertension standpoint, right? So if we jump out to the 14-month, we see that distribution spreads out, right? It drops down and really spreads, okay? So something is causing these animals to increase their PAP across the feeding period, right? Do we know what that is yet? I don't think so. At least Tim doesn't, because he's still out there papping for us every time. And I don't know if he'd be as willing if we knew exactly what was causing this, right? So what does this mean in relation to heart score, right? So how does this translate to heart score? Okay. So again, on the left, we have the 14-month, or the 9-month PAP. On the right, we have the 14-month PAP. And then on the left of each of those blocks, it's ordered heart score 1, 2, 3, and 4, okay? So if we look down here, there really is no difference. Oh, I lost my pointer. Maybe I can draw right here. So if we look over here, there really is no difference between all of the different heart scores at harvest and their 9-month PAP, right? Which is what that other distribution showed us. But if we sit there and we break the PAPs up by their 14-month PAP up according to their heart scores, we see this big exponential relationship, right? And that's kind of concerning. It's concerning because what is causing this to happen, right? Because we're not at elevation, so there is no really lack of oxygen like Dr. Holt was saying earlier that's causing this increase in PAP. So something else must be going on causing these animals to become hypoxic, right? So if we think about this though, as you go from a 1 to a 2 to a 3 to a 4, if Dr. Holt told us, if what Dr. Holt told us was right, we expect those higher peaks, right? We expect that higher peak in the 4. Higher PAPs mean greater heart failure, incidence of heart failure. Okay, so just real quickly, some results. We've got healthy versus unhealthy, and the differences we saw in the healthy versus unhealthy, we saw a difference in 14-month PAP, and these are all highlighted in yellow. We see a difference in marbling, so the unhealthy hearts have higher PAPs at 14 months of age. They have lower marbling scores. They have higher hot carcass weights, and then they have a lower feed to gain ratio, so they're more inefficient. Okay. If we think about these per individual score, I'm going to skip over this just due to the fact of time because this is really showing what I had on the previous slide. Is as heart score goes up individually, we see PAP going up. But if we look at some of our traits, so the top there is marbling score. We see a linear, it's a little bit curvilinear. You could probably test that, but you can see a decrease in marbling score as we go across from a 1 to a 4. Then if we look at that bottom graph, that's carcass weight. So you see there's a curvilinear relationship. Carcass weight's increasing up to a point to heart score 3, and then we're seeing a drop off as we get to those 4s. So that kind of brings back the question that Dr. Vanderlei had on, is this causal or casual relationships, right? Is one causing the other one, or is one a result, or vice versa, okay? We don't know. And then just on a subset, because with that carcass weight, we've got this other gain in the feeding period. So when these groups of animals were in the feed intake unit, you see the gain across that time, and you see the same relationship with carcass weight as you do with gain in the feeding period. So that nonlinear or that curvilinear relationship. So that might help explain why some of those correlations are so low, because those correlations are looking at linear relationships between the two traits. Okay, so let's get into the genetics here real quick before I run out of time. So we have a heritability heart score of 0.28, okay? So this was done on a sire model. This is a small data set, but we have pairwise correlations between a number of these. So 0.28, that's very similar to what those PAP heritabilities that Dr. Enns was presenting earlier. If we look at the genetic correlations between traits, right? So if you go all the way over to the right in that other highlighted box, we've got back fat, ribeye area, and marbling. We see the strongest genetic relationship in that group is with ribeye areas. So larger ribeye areas are going to have higher heart scores. Animals with larger ribeye areas are going to have higher heart scores. Then if we come down even further, back to hot carcass weight, that's the strongest relationship. So we have a 0.63 genetic correlation between heart score and carcass weight. So meaning, the way I like to interpret that is if you think about two traits that have a set of genes, and the genetic correlation is measuring the percentage of genes that influence both traits, right? So we would sit there and think that, okay, 63% of the genes that influence hot carcass weight are also influencing our problems with heart score. So this kind of starts to paint a picture, right? We've got heavier carcasses, higher heart scores, bigger ribeye areas, maybe a little higher heart scores. There's some contradictory information here with regard to phenotype, but there's some other influences that are going on there that might cause that to those relationships to erode when we look at phenotypically. And then again, the PAP, that 0.94, that's the genetic correlation with 14-month PAP and heart score, which is real. They were measured really close together within less than a month. So that relationship is genetically extremely high. So just some highlights really quick. Heritabilities, 29% heritable. We really don't see, at least in our work, we're not seeing heart scores of 5 in the plants. 14-month PAP in our data is really the one that's showing a relationship with heart score. Higher PAPs are indicating higher heart scores. Phenotypically, we're seeing increases in heart score associated with decreases in efficiency and decreases in certain carcass characteristics, primarily carcass weight and marbling score, even though the genetics is opposite of what that's telling us. And then if we see PAP increasing across the feeding period, again, we have the heritability there, but we're seeing antagonistic genetic correlations with primarily the carcass traits. The biggest one is carcass weight. The next largest one is ribeye area, and then we've got some really weak genetic correlations between back fat and marbling. And there we go.